My name is Shaimaa Bakr, and today I'm going to talk about identifying key multifunctional components shared by critical cancer and normal liver pathways via sparseGMM, a method I developed. Many diseases have significant genetic underpinnings that determine the underlying pathology. In cancer, genetic and epigenetic events in certain genes cause dysregulation of biological pathways and downstream changes in gene expression. Reverse engineering gene regulatory networks is a challenge and opportunity to improve our understanding of disease and to identify new drug targets. With the increasing availability of multi-omic data, there is a rising need for statistical models that can integrate such data. Here we develop a method for gene regulatory network learning from transcriptomic data using a Bayesian approach and apply it to normal and liver cancer data. Figure 1 shows an overview of the study in the sparseGMM method. Given bulk RNA-seq data, sparseGMM uses latent variable modeling to learn Gaussian mixtures with sparsity constraints on the coefficients of regulators. SparseGMM is initialized with random assignments of genes to modules, and the learning takes place through a two-step expectation maximization algorithm. To measure robustness of modules and to filter for robust modules, sparseGMM is run several times, generating multiple gene networks. Next, robust modules are screened by running a community detection algorithm. This groups together robust modules that are consistently discovered in every run. To functionally annotate the robust modules, we apply gene set enrichment analysis to MSigDB gene collections. Finally, to validate regulators of robust modules, we use publicly available perturbation experiments that identify experimental targets. We applied sparseGMM to detect data from healthy liver tissue as well as hepatocellular carcinoma samples from TCGA. We combined normal and cancer modules to uncover shared biology between two types of tissue. We discovered 72 communities, 22 of which were identified to be robust using community detection. The definition of robust community is one with a Jacquard index greater than or equal to 0.7. The analysis of healthy tissue points to sparseGMM identified genes as potential transcriptional biomarkers of hepatic metabolism and validated BDH1 and HADH as regulators of this process. Analysis of HCC lesions shows that the clotting system and the antigen presentation processes are robustly regulated in HCC and are driven by important regulators such as hepatocyte nuclear factor 4 alpha and PDCD1-LG2. The combined analysis of both tissue types highlights the importance of angiogenesis for tumor growth and confirms important regulators such as protein C receptor. Single-cell evaluation of immune function modules demonstrates the robustness of these communities and the ability of sparseGMM to decouple distinct myeloid and lymphoid processes. Generative models allow us to calculate the probability distribution of target genes in the form shown. The computed probability distributions can be used to calculate entropy. Entropy is a concept from information theory used to measure uncertainty and is inherent to the probability distribution of the random variable. High entropy genes have high uncertainty in their assignment to gene modules. As Figure 5 shows, target genes in heterogeneous cancer tissue have higher entropy than normal tissue. High entropy can be used as a proxy of membership of multiple biological processes and can unveil hidden elements of crosstalk in cancer. Functional crosstalk is an interaction between two pathways belonging to different cell processes. High entropy genes are more present in a few subnetworks such as p53, NF-kappa B, TNF-alpha, interferon gamma, and estrogen. To conclude, sparseGMM is a Bayesian approach to module-based gene regulatory network inference with sparsity constraints. sparseGMM captures biological processes in liver cancer and normal liver, as well as shared processes that occur in both tissue types. sparseGMM uncovers multifunctional genes with potential involvement in functional crosstalk that can serve as drug target candidates and improve our understanding of cancer biology. Finally, experimental work is key in validating findings of sparseGMM in HCC. Thank you.